we have gotten our visual information to the visual cortex, and now we are going to go beyond V1. Well, how did we get here? If you don't know your talking heads, that is a shout out to the talking heads. Or I'm sorry, it's just talking heads. There's no the. We talked about the stimulus, the electromagnetic energy, that is light. It hits the photoreceptors, our sensory neurons, that then send the information to the bipolar cells, which send the information to retinal ganglion cells. I'm ignoring that crosstalk or the lateral perpendicular talk of the horizontal and amacrine cells there, but this is our pathway. Photoreceptors to bipolar cells to retinal ganglion cells. We're still in the eye. The retinal ganglion cells, their axons make up that optic nerve, that nerve fiber, that particular cranial nerve that travels to the brain, and it brings the information mostly to the lateral geniculate nucleus, but also some information goes to the superior colliculus in the midbrain that helps us to figure out where we want to move our eyes based on that information, as well as information from other sensory modalities. The lateral geniculate nucleus, that LGN, which you can also always just call it LGN when you're writing anything out, I'll know what you mean. It sends it's the thalamic nucleus that is doing that summarizing, sorting, filtering so that the brain can find the important information, and then it sends that information on to the striate cortex, which we've also called the primary visual cortex and V1. So where does it go from here now for higher level processing? There are two primary pathways leaving the striate cortex. One is going dorsally through the brain, where dorsal is going up towards the top of the brain. They often say to think of the dorsal fin on a shark. For four-legged animals, that is the back, but because we are two-legged animals, we have our dorsal is towards the back, but it's also towards the top of the head, if you kind of imagine that back coming around it up towards our head. So we're going to take that information dorsally through the parietal lobes, and that's going to be the pathway for understanding where things are in space, helping us to navigate in space. And we might talk about it as a how pathway as well, or we will talk about it as the question of where or how do we navigate through space. And then the other pathway is the ventral pathway. So ventral towards the stomach or in the brain, that's down in the brain, going through the temporal lobes. The temporal lobes are really helping us with our object recognition. They're often called the what pathway. If I introduce how we discovered these pathways, I'm going to talk about this early study from Leslie Ungerleider and Mortimer Mishkin, if I'm saying everybody's names right. They did an ablation study, which is a lesioning study, where the way these studies work is that they first test the animal's capacity. They train the animal, measure perception, measure what they're interested in. Sometimes it is cognition, but in this, for us, it's perception. They remove a particular area or lesion and destroy a particular area and leave the rest of the brain intact. And then they retrain the animal to determine which of the perceptual capacities, again, in our case, perceptual capacities still remain. And so whatever that animal is then missing or unable to do, we're going to say that that brain structure was crucial or played a role in that particular task or perceptual capability. So in the Ungerleider and Mishkin study, they presented the monkeys with two tasks, or got them trained up on two tasks to make sure they could do object discrimination, where they're telling the difference between a cylinder and a rectangular object, and the food is always next to one of those, and they get trained up on that object recognition or object discrimination. And the other is a landmark discrimination problem. So here's this landmark, and the food well is going to be next to that landmark that's telling me where in space it is. So if we look at these ablation studies, if you see the brain down there, what they're showing is that little purple area is the area that is ablated when they are ablating the temporal lobe. So no temporal lobe, they're unable to do that object discrimination or being able to choose the correct object. This is the ventral pathway. This is the what pathway. And if I put this all together as simply as possible to just connect those words together, it's the temporal lobe that is the what pathway for vision. The temporal lobe is doing other things, but that's the what pathway for vision. It's letting us do our object recognition. This is the ventral pathway through the brain. We look at that, the ablation, and you see the purple area now that they've taken out in this area of the parietal lobe, and now it's the landmark discrimination. So here's this well that is a landmark showing me this is where the food is going to be, or where. So this more where or navigation kind of information. And now if the parietal lobe is taken out, the monkey cannot do the task. And this is the dorsal pathway. I'm going to do the same thing where I just put these together. We're going from the striate cortex to the parietal lobes. This is the where pathway, the dorsal pathway. And so again, just putting these words together to, these are the pieces and parts that you kind of need to connect and associate with each other. If we look at where information is coming from back in the occipital lobe, our primary visual receiving area, it is going along two pathways, the ventral pathway to the what or temporal lobes, the dorsal pathway to the where or parietal lobes, and it is often discussed as a where or a how, since there is this aspect of where things are, but then there's also how I'm navigating or how I'm using that environmental information. And to discuss the specific pathways, we're going to start with the dorsal root. So this question of should they be called what and where, or should they be called what and how? This was asked by David Milner and Melvin Goodale as they were working with people who had brain damage and were having difficulty. They specifically worked with this patient DF, and she had difficulty, or she had damage to the parietal lobes and had difficulty with this where, but maybe they thought of it more as a how component because of the tasks they were asking her to do. So if we think about this dorsal stream, the where, that they called it a where, the how pathway suggests that it involves taking action within our environment, which necessarily requires a where component. I used to ask classes early on, and then there's really no answer to this, so I stopped asking the question, even though the whole thing is to have this, whole idea behind the question is to have you think and reflect and have this kind of thought process of how would we be able to disentangle these things, this how from the where things are in the environment. So we have the where component, but the taking action also involves physical interaction with objects in the environment. And I am going to back up just a little bit. So again, they were looking at a patient who had some brain damage, and this kind of evidence is neuropsychological evidence. So the evidence comes from neuropsychology, or the study of behavioral effects because of brain damage in humans. And one of the important distinctions or definitions to be made in neuropsychology is this distinction of the definition of dissociation, and then the distinction between single dissociations and double dissociations. So a dissociation is a situation where one function is absent while another function is present. Okay, well, I'm going to distinguish between the single dissociations versus a double dissociation. So a single dissociation, we have a patient, Alice. This is an example from the book. She has damage to her temporal lobe. She has difficulty with naming objects, but no difficulty with locating objects. So now, because we can see this what pathway causes the damage to naming objects, but she's still able to locate objects, we can see that those two functions naming and locating, involve different mechanisms or different areas of the brain, different mechanisms in the brain. We don't know if they operate independently of each other or truly independently of each other. What we know right now from Alice is that I don't need to be able to name objects in order to locate them, but it could be that in order to name objects, I have to figure out where they are, right? I don't know that they're truly, completely independent of each other. This is only a one directional kind of, we know they're different mechanisms. For a double dissociation, we need at least 2 patients to see this. So we have Alice with her temporal lobe damage, having difficulty naming but not locating. Well, let's take Bert. Again, these are just examples from the book of how these dissociations work. Bert has damage to his parietal lobe. He has difficulty locating objects, but no difficulty in naming objects. And now we can conclude that these 2 functions have different mechanisms in the brain, different areas of the brain are responsible, and that these mechanisms of the brain can operate completely independently of each other. So as we saw with Alice, you don't necessarily need to be able to name something in order to locate. But now with Bert, we also see you don't need to be able to locate an object to say where exactly it is for someone to point to it and you to say, that's a cup. Okay, so we can see that these are fully independent of each other, or that they can be. I don't want to state that too strongly because in a brain that is not damaged, these regions are definitely communicating with each other. And I've actually had a student do their project talking about looking at the dorsal pathway and how it is involved in just face recognition, that we do use movements of the face and so forth. And so they can operate independently of one another. And we see these double dissociations in many of our functions and tasks that we do. So a lot of our memory systems are fully doubly dissociated. Language systems can be doubly dissociated as well. If we get back to Milner and Goodale, so they were looking at this patient DF, and she had damage to her visual pathway, and you can see that in the top task, what she's supposed to do is match the orientation of a card held in her hand to different orientations of a slot. And you can see the control people are people without any brain damage, and the slot is vertical, and they're showing that we're all right on or really close to being completely vertical. Nobody's off by very much. We look at DF. She is all over the place, right? She's oblique. She's actually completely horizontal. She has no ability to match that orientation of the slot. But then they ask her to, well, place the card in the slot like you're putting it through a mailbox. The controls, again, very good at matching to get to the vertical and then placing the card in through the slot. So is DF. So asked to perform this task, and she's fine. She's right on. She looks like a control person. So we can conclude that we have one mechanism for judging orientation along that what pathway, because that kind of thing is going to tell us, right, about objects as well and the what's of things. But then we have another mechanism, a different mechanism for coordinating that visual information with action. As we just saw with DF, we have one mechanism for judging orientation, a separate mechanism for coordinating vision and action. That's what we can tell from that single dissociation. Do we know if these mechanisms are independent of each other? These are my green questions for you to think about, so you can pause. But my next question of how do we find out suggests that the answer is no, right? We don't know that. How do we find out? Again, I'm going to recommend it that you pause and think if you don't already just have it on the tip of your tongue. We need the other person, right? We need another patient. We need somebody else who has damage to the parietal regions, I'm going to say, who has the difficulty of coordinating the vision and action, but when they're doing the static judgment of orientation, they can do that just fine. Right? We need the opposite. We need to see the opposing functions being intact and impaired. So there are people who can judge visual orientation, but they can't complete the task that combines the vision and the action. So they're not able to coordinate vision and action, but they can judge visual orientation. And again, this is damage to the dorsal stream causing this. And it does look like these mechanisms are independent of each other. And my question of, does this sound more like a how, right? How much of this is a where pathway versus it's really coordinating vision with action. So the dorsal pathway gets discussed in, it actually gets discussed in all these ways, as a where pathway, as a how pathway, as an action pathway. We can also see evidence from people without brain damage. So if you look at the middle, I usually start in the middle with B, you can see that the line 1 is longer than line 2. But if we put it into A, right, where we have these depth cues that suggest that 2 is farther away, well, if something is about the same size on the retina, but it's farther away, it must be larger, right, than if it's, so usually things, it's not if I'm close to you, I'm huge on your retina. If I walk away, I get smaller and smaller on your retina. I take less and less of that visual space or visual angle up. And so line 2 appears to be a little bit longer than line 1, even though in reality it goes the other direction. And you can see, so what they did with the people is they put these little electrodes on their fingers to measure the distance of the fingers. And when they're just judging the length of the line, you can see that they're judging 1 as being smaller than 2. Again, that depth cue works, and 2 appears to be larger. But if they go to reach for the line, they change their fingering so that it really nicely matches the actual length of the line. So 1 is just slightly longer than 2, and our fingers go slightly farther apart for 1 than for 2. That was the where or how pathway, but now talking about the what pathway, this ventral route. We do want to get beyond those feature detectors. We want to put those features together into objects. And Charles Gross and his colleagues, actually back in 1972, were looking in the inferotemporal cortex, or the IT cortex of monkeys, and they were, guess what, looking at what looks like a center surround kind of receptive field, and looking at basic features, and they were finding almost no activation of these neurons, of these single cell recording of these neurons in the inferotemporal lobe. And so they weren't sure what these neurons responded to. And then one day when the monkey was hooked up still and they were measuring from the appropriate neuron here, the experimenter reached across and such that their hand created a shadow on the screen and the neuron fired. And so they changed their materials. They went and they, you can see that these particular neurons they were recording from responded maximally to things that looked like they could have fingers or hands. It responded absolutely maximally to what looks like a monkey's paw. And as we get farther and farther away from what looks like a hand with those digits, we have less and less activity from those neurons. So we are beyond feature detectors now, putting them back together to be objects in the world. Here in a 1991-1995 set of experiments by Tanaka et al., they found what I call, don't know if they called it this, I keep meaning to go look at the research, the lollipop neuron. So this neuron responds, and that neural responding at the bottom, we have the highest amount of responding to the lollipop, almost no responding to just the stick, very little responding to just the circle at the top. And I can change the shape of the lollipop to a square and it responds, but not quite as much, which is why I call this a lollipop neuron. I am almost to my 20-minute mark, so it is a good time to take a little break, stretch, whatever you need to do between these videos. We are in the middle of the what pathway, that's this ventral route through the temporal lobes that really help us with our object recognition. We are going to continue to talk about how we go beyond feature detectors in the brain to put that information together into something the brain can understand and categorize as real objects in our environment.